Vishmaya. Is my screen visible?
Yes, sir.
Yeah, okay. So I'll give the revision for third unit. Um, so what I'm going to do is I'm going to give idea about how to prepare the third unit. Okay. So you know the question pattern, right?
Yes,
Yeah. The
sir.
question pattern is you'll be having uh, 10 2-mark questions, 20 marks. Then you have to write 4 out of 6 15-mark question. Okay. 60 marks and 1 20-mark question compulsory. 1 into 20 is 20, so total 100 marks. Okay. So I gave revision for the first and second unit. I hope you know all the materials are available for the first and second unit. I have to upload only one material for the first unit on importance of stereochemistry in drug design, right? Am I right? You people can respond. Am
Yes,
I
sir.
audible? Yeah. Okay. So the third unit is on receptor, ion channel, and enzyme. So three major topics there: receptors, ion channel, and enzyme. Okay. So all this, partially you might have studied in your medicinal chemistry. Also, I teach you this, receptor, ion channel, and enzyme. So how to prepare for this? First, you have to prepare in three aspects. Okay, the first one is in terms of structural aspect. Secondly, you have to prepare in terms of the functional aspect, and third thing is you have to prepare in terms of uh, pharmacology aspect. Okay, so these are the three aspects in which you have to make preparation for this unit, right? So three things you have to prepare. One is the receptor. Second one is the ion channel. Ion channel is also kind of a receptor. Okay, then the enzymes. Okay, so let me start with the enzyme, right? So because that's very simple, right? So what are the things we have to prepare for? Enzyme. Okay, so the first thing is enzyme kinetics. Okay, so you have to prepare like what's the function of enzyme? Okay, general function of enzyme, and then enzyme inhibition. Okay, enzyme inhibition. Then type of enzyme inhibition. Okay, so we have three types of enzyme inhibition. The first one is the competitive inhibition. Second one is the non-competitive inhibition, and the third one is uncompetitive inhibition. And this is very important for the examination point. Okay, so we can expect one essay type question out of this, either competitive inhibition or non-competitive inhibition or uncompetitive inhibition, right? So what the examiner may expect is with clear illustration, right? So if you refer the lecture I have given for inhibition, so you can find some simplified illustration for all the three type of inhibition, right? So if it is a competitive inhibition, okay, so the substrate that binds to the enzyme where it forms a complex. Okay, substrate enzyme complex. Okay, so which eventually lead for the product formation, right? So either the substrate it can go and bind, or So the inhibitor, it can go and bind. So both of this going to compete for the same binding pocket. Okay. So this you have to explain with the help of a very neat graphical illustration. Second one is non-competitive. Okay, so in case of non-competitive inhibition, the binding site for the substrate and the binding site for the inhibitor, they're not the same, they're different, and the substrate and the inhibitor, it's not going to compete for the same binding site. So how we can make the representation, you can make representation like this. Okay, so it's a very simple graphical abstract of non-competitive inhibition. So this is the substrate molecule, and say this is the inhibitor molecule. Okay, so this is the main binding site. We can call it as the active site, where the substrate goes and bind, where the substrate molecule will get converted into the product molecule. So this is inhibitor. Okay, so this inhibitor has a different binding site. This time it's not the same active binding site, so it can go and bind to the site called as allosteric site. Very important, you have to remember this. So this site is called as allosteric site. Enzymes may have many number of allosteric sites. Okay, so because if the substrate alone goes and bind, it will be converted into product just like this. Okay, but if the inhibitor binds to the allosteric site, what it can do is like it can alter the conformation of the binding pocket of the active site. Because of that, the substrate molecule may not able to go and bind, and it may not able to get converted into the product. Okay, so equation wise, substrate plus enzyme. Okay, so am I audible now?
Yes, sir.
Yes, sir.
Yeah, I think I got disconnected. Okay, where I stopped, I explained so many things.
After allosteric site, you were
Okay, so
explaining.
I think this is what you had to cover for the non-competitive. Okay, so the third type is is very uncommon, uncompetitive, in which the inhibitor that binds to the enzyme, okay, which lead for the formation of enzyme inhibitor complex, okay, to which the substrate molecule it can go and bind. Okay, eventually it will not lead for the product formation, right? So for the examination point, again I repeat, this is very 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 important, right? So we can expect some question out of enzyme inhibition. So either a competitive inhibition or non-competitive inhibition or uncompetitive inhibition. Okay, so that's what you have to prepare in this. So coming to the transition state analog. Okay, but this you might study in your medicinal chemistry, right? Like during the conversion of substrate molecule to product, right? So substrate which binds to the enzyme, okay, and the enzyme which convert the substrate molecule into product. It's a very normal thing. That's the main job of the enzyme, right? So if this is the energy of the substrate, and if this is the energy of the product, right, so always the energy of the product should be lesser than the energy of the substrate. So for the conversion of substrate into product with the help of enzyme, okay, directly this conversion is not possible. So what should happen is that it should reach some energy. Okay, this energy is called as the transition state energy. Okay, this energy is the transition state energy, right? So for the conversion of the substrate this is the energy difference between the substrate and the product. Okay, so the product is having lesser energy comparing to the substrate. For this conversion, the system has to spend some energy, and that energy is called as the transition state energy. Understandable?
Yes, sir.
Yes, right. Okay, so so this transition, so at this point the substrate molecule will be having some conformation. Okay, very important to understand. The substrate molecule will be having in some conformation in this transition state transition state. Okay, so that conformation is what is called as a transition state conformation. Okay, so here if you design any drug molecule that mimics the conformation of the transition state of the substrate, then that is what is called as transition state analog. Is it understandable?
Yes, sir.
Okay, so if we design any new molecule that is having a very similar structure, very similar conformation, very similar energy like the transition state of the substrate, those mimics are what are called as a transition state analogs, right? So this is very special kind of designing in which they will try to mimic the transition state, because the transition state of the substrate, okay, its structure is very crucial for the conversion of the product. Okay, that is what everything is, on my slide you can go through that. So what is meant by suicide substrate? We cannot expect any essay type question out of suicide substrate, right? So what is suicide substrate? So initially the drug molecule will be in its inactive form. Okay, initially it will be in its inactive form. Okay, so I just go for a small illustration here. Say for example, the molecule is initially in its inactive form. So this is the drug molecule. This is the inhibitor which is in its inactive form. So once if it enter into the binding site of the enzyme, so what will happen is that the inactive molecule will be converted into active molecule. Say this is active, right? So actually this is something very similar to the induced effect. Okay, the main difference is, so now what happens that, so this inhibitor will form a covalent bond interaction with the substrate binding site. Very important, it makes a covalent bond interaction with the binding site. So because of the formation of covalent bond, now this molecule cannot come out. Okay, we know that the relationship between the drug molecule inhibitor and the enzyme or any target is that drug molecule goes and bind to the receptor, where it makes drug receptor complex, but it is going to be reversible in most cases. Okay, but in very few cases the same reaction is going to take place only in the forward direction, not in the reverse direction, which means that in the second example it is not reversible, it is irreversible. So what is responsible for irreversible formation of complex between drug and the receptor molecule or the enzyme molecule? It is because of the formation of covalent bond. So it makes covalent bond here, and that is why it is called as suicide inhibitor. Is it understandable? Those are called as suicide substrate. Is it understandable?
Yes, sir.
Okay, so these are the main things that you have to study for this specific part, right? So coming to the first part, first thing is you have to study about the general structures of, even before the general structures, first you have to study about classification of receptor. If you remember, in one of my presentation I discussed about the classification of receptor. So we can classify the receptors into different subtypes. Okay, superfamily type 1, superfamily type 2, superfamily type 3, and superfamily type 4. Okay, superfamily type 1, type 2, type 3, type 4. So superfamily type 1 is ion channel. Superfamily type 2 is GPCR. Type 3 is tyrosine kinase linked receptor, and type 4 is the nuclear receptor. Okay, so you have to give one example for what is the corresponding ligand molecule which can activate the receptor. Okay, so these are the things you have to start preparing with, right? So your preparation should start with classification of receptor. That's the first part, right? So coming to the second part, you should have very good idea about the basic structures of superfamily type 1, type 2, type 3, and type 4. All right, so because there's going to be a structural difference between type 1, type 2, type 3, and type 4. So structure in the sense you have to discuss the architecture of type 1, type 2, type 3, and type 4. So if you take superfamily type 1, okay, it will be having more than one subunit. Okay, so normally they used to have 4 to 5 subunits, membrane spanning domains. If you take superfamily type 2, it is used to have a very constant number of membrane spanning helix, right? So it's going to have 7 transmembrane. So superfamily type 3 is going to have only one transmembrane. Superfamily type 4 is specifically very different type of superfamily because it is not present in the membrane, it's not embedded in the membrane, it is present inside the nucleus. Okay, so then you have to tell how many number of helices, whether the N terminal and C terminal, what is the localization of N terminal and C terminal, where it is present, whether it is present in the extracellular region or intracellular region, because in some cases both the N terminal and C terminal used to be present in the extracellular region. Some cases N terminal will be present in the extracellular region, and C terminal used to be present in the intracellular region. Those are the things you have to study in terms of structural aspect. Is it understandable? So coming to the third part is the mechanism, right? Mechanism of superfamily type 1, type 2, type 3, and type 4. So you have to give graphical illustration of the mechanism of superfamily type 1, type 2, type 3, type 4. So coming to superfamily type 1, the mechanism is like here also you have to discuss the general structural feature of superfamily type 1. So coming to the mechanism, so you can take acetylcholine receptor. Okay, so used to have 5 subunits. Okay, so 2 alpha subunits, 1 beta, 1 gamma subunit. Okay, so for the activation of this, okay, superfamily type 1, basically they are ion channels, right? So you have to discuss a few words related to what are the main functions of ion channel, okay, in normal cell. Because if any molecule is in its ionized form, it cannot transport it inside the cell with the help of passive diffusion. The best possible mode of entry of those molecules into the cell is going to be ion channels, right? So the primary job of superfamily type 1 is they are ion channels. So for the activation of this acetylcholine ion channel, we need 2 molecules of acetylcholine, and you have to discuss different domains, different subunits which are present in this, right? Those are the things that you have to cover for superfamily type 1. So superfamily type 2 is you can give example as GPCR, the mechanism of G protein coupled receptor. The general idea of GPCR is that so it is used to have a very specific domain called as G protein binding domain. So let me give a graphical illustration of this, right? So GPCR. So So here there is going to be some domain called as G protein domain. Okay, so where the G protein will come and bind to this, right? So what happens initially, the ligand will come and bind in the extracellular region. This is the extracellular region. Ligand will come and bind in the, so ligand binding domain may be either present here or somewhere here. So where the ligand should come and bind. So because of the binding of the ligand, it activates intracellular part domain called as G protein. So now the G proteins are attracted towards this. Okay, basically the G proteins are made up of three subunits, alpha, beta, and gamma subunit. So to this alpha subunit, there will be one, GDP, right? So this will be attracted towards this. Okay, because by after the binding of the ligand molecule, some conformational change will take place here. You can refer my lecture on this. So that attracts this G protein. So G protein will go and bind. So because of the presence of this GDP, it is highly unstable or less stable. Because of this alpha subunit with this so this GDP will be first converted into GTP. Okay, alpha, beta, and gamma subunit. So because of this conversion, this subunit is going to be highly unstable and dissociate from the alpha and beta and gamma subunit. So this alpha subunit along with the GTP. So this is called as the now kind of another messenger which can go and activate any other ion channel, or it can activate or inhibit any other enzyme. That's the general mechanism of GPCR, right? So coming to the third super type, super family type receptor, it is a tyrosine kinase linked receptor. The specialty of tyrosine kinase linked receptor is that this is the only family of receptor which is used to have two type of properties. One is the receptor property, another one is the enzyme property. Okay, so which means that it can do the function of both the receptor as well as the enzyme. So the mechanism is that when the ligand bind to this tyrosine kinase linked receptor, so dimerization will take place immediately. So two proteins will come and join. So it is used to have a intracellular domain called as SH domain. Everything I explained, it just I am giving the summary of what the examiner may expect in the answer script. So this domain, right? So here you may have many number of tyrosine amino acid. So because of the dimerization, this tyrosine amino acids will be phosphorylated. Okay, so all the OH will be converted into P because of the phosphorylation, which attracts the SH domain and which can activate the transcription and other factors, right? So this is the general mechanism of type superfamily type 3. Superfamily type 4 is as it is present inside the nucleus, right? So its main job is that the specialty of superfamily type 4 is first it is present inside the nucleus. Okay, this is only family of receptor which present inside the nucleus. The second important point is that they used to have a specific domain and motif. Okay, called as DNA binding domain. Okay, so motifs like zinc finger, these are very common in DNA binding protein molecule. So they used to have that. So because of that specific domain, the DNA can go and bind and it can regulate the transcription and translation processes. Understandable? So
Yes,
these
sir.
are the things. This is the way how actually you have to prepare. It's not lengthy unit, right? So you can finish comfortably within a few hours, right? Very comfortably you can finish. So you should know at least a few examples you can explore on your own about what are the disease which are associated with malfunctioning of ion channel or malfunctioning of any superfamily of receptors, right? So that you can include with this answer. That is all about the third unit. Understandable? So if you have any question, I can answer now. What I said is how to prepare this unit for the examination. If you have any queries or if you have any doubt, I can clarify that.
Okay, so on the 20, and that's less than 20, which second group and this stage
No
2.
questions, sir.
Okay, so at any point of the time, if you have any question or if you need any help, right? So in terms of material or any video, if you need additional, just write an email to me or put it on the WhatsApp. Okay.
Okay.
Okay,
So
sir.
till the exam time, till your date of examination, I'm ready to help you in all possible ways. Okay.
Thank you, sir.
Yeah.
Yes,
Okay.
sir.
Then we can wind up the session. Thank you.
Thank you, sir.
Yeah.
Thank you, sir.
Yeah.